Hi ladies, today I'm going to be giving you some notes on organic molecules. So what does it mean to be organic? Well, organic essentially means anything that's made of carbon. So some of the examples of molecules that are made of carbon would be the carbohydrates, proteins, nucleic acids, and lipids or fats. And you're probably hearing some of these words and thinking, hey, I eat those on a regular basis. When I look at the food labels for anything that I'm eating, I'm typically eating organic molecules. And the reason that you eat organic molecules is because they are a great energy source. Okay? There's a lot of energy stored in between those bonds, and when those bonds are broken, your body can get a lot of energy that it needs in order to live. So why is carbon such a great backbone? In order to think about that, you have to think about the structure of the carbon atom. So if you look up here, this carbon atom has 4 valence electrons, which allows it to form 4 bonds with 4 other atoms. Looking here, you can see that there are many carbons that are linked together in a long chain. Okay, this is a fatty acid, so when we talk about lipids a little bit more, we'll see this again. But you can see that between each one of these carbons, there's a bond, and when this is broken or when your hydrogen's removed, okay, later on, this is going to release a lot of energy that your body can store. Okay, forming 4 bonds forms a very stable molecule as well. So in addition to storing a lot of energy, okay, it's also able to be structurally very stable. So these molecules aren't explosive or anything like that. Okay, so a couple of the terms we're going to need to know and be comfortable with today are monomer and polymer. So let's just focus on this picture down here for a second. So monomers essentially are like subunits, repeating little pieces, building blocks that make a polymer. So you put a bunch of monomers together, you get a polymer. So a good way to think about monomers and polymers is like Legos. So you know those little 4-piece Lego blocks or whatever little tiny Lego blocks? Okay, if you take those and you put them together to maybe build like a castle or build a house, okay, you're taking those tiny little Lego blocks, which would be the monomers, okay, repeating subunits, and you're making a giant structure. That would be your polymer. Okay, you can't break down a monomer into anything smaller. I can't take those tiny little Lego pieces and break them down and make them into a different shape. Okay, that's another thing that monomers have in common. Okay, they're a repeating subunit, and that little building block can't be broken down any further and be considered a monomer. So when we're making and breaking polymers, we have 2 different processes. Dehydration synthesis, so making a polymer by removing water, okay, and hydrolysis, okay, breaking a polymer into monomers with water. So a good way to think about things in biology is to think about root words. Hydro right here, water, lysis means to break. You're going to see that several times. So you see here I have an H, an OH. These can be removed through dehydration, okay, removing water, to synthesize a polymer. Okay, each one of these little circles represents one of the monomers. Okay, here, circles represent monomers. Adding water, okay, you add that hydrogen back here, you have that hydroxyl group right here, and that will break the bond and make a monomer over here, and then leave the rest of the polymer over here. And this applies to every single organic molecule that we're going to be talking about. Okay, they're not going to be these little circles, okay, they're going to be specific different monomers for different types of organic molecules, but the same process holds true for every single organic molecule. This is how they're made, this is how they're taken apart. First one I'm going to talk about are carbohydrates. Carbohydrates have monosaccharides as their monomer. Okay, when you link monomers together, you're going to build a polysaccharide. Okay, so monosaccharides is the monomer. You put them together and you can make a polysaccharide. So it's the same word here, but just poly in the front. Basically, carbohydrates are for energy, okay, short-term energy. Okay, one of the most common monosaccharides is actually glucose. So you can see this long chain of glucose here. Okay, if you connect the 2 sides together, you can make the ring form of glucose linked together. It can be drawn here 3-dimensionally. You might even just see something very simplified like this to represent glucose. This is short-term energy. So if you think about why you eat sugars and why you eat these carbohydrates, they're for energy. they can be put together when they're turned into a polymer. You can actually form a glycosidic linkage. So if you look down here at this molecule right here, you see this OH here, you see this hydrogen here. If these are removed with an enzyme, which we will talk about later, this is the dehydration part, okay, to synthesize a polymer. Okay, this is this molecule is made up of 2 glucoses linked together with that glycosidic linkage, and this is what we call maltose. If you've ever heard of sucrose, sucrose is actually a disaccharide, so 2 monomers put together, and it's a combination of glucose, and then the second sugar would be fructose. So table sugar, if you put that in your coffee in the morning or something like that, that's a combination of glucose and fructose. It's a disaccharide, so 2 sugars put together. If you think about the properties of this molecule, sometimes you want to think about if it will dissolve in water or not. So this molecule right here has a lot of oxygen. You remember we talked about oxygen being very electronegative, which means that this molecule will be slightly polar, which means it will dissolve in water, which is also slightly polar, like dissolves like. Okay, and you can look at the different elements. Typically the elements in the carbohydrates are going to be carbon, oxygen, and hydrogen, usually in CH2O, okay, raised to whatever n power. Okay? And we'll talk about that a little bit more when we review this. The last thing I'm going to talk about are the different types of polysaccharides. So if you look here, the different types of polysaccharides that are carbohydrates are starch, glycogen, cellulose, and chitin. Starch is a polysaccharide that's made up of, these are all made up of little glucose monomers, but the forms that they take are slightly different. Okay, so this one has more of a helical structure. This one, glycogen, is used for branching. Cellulose and chitin are very linear and very strong the way that they're held together. Starch is basically a plant short-term energy source. You can also eat it as well. Glycogen is your short-term energy source. We're going to spend a lot of time talking about glycogen whenever we talk about insulin later. We're also going to look at cellulose a little bit. Cellulose is a plant structural structural carbohydrate. And chitin is the animal structural carbohydrate. If you ever see you know how insect exoskeletons crunch, okay, or plants have that firm structure, this is because of the cellulose and the chitin. Okay, and all of these are made up of glucose monomers. Next are the proteins. The protein have a monomer that are called amino acids. The amino acid monomer looks like this. It has a central carbon, it has a carboxyl group, it has an amino group, and then it has a hydrogen and a variable R group. Okay, the R group represents different variation between these monomers, so I'll show you that in just a second. In order to link 2 amino acids together through dehydration synthesis, you're going to form a peptide bond. Okay, remove water to make a peptide bond. These are used in all sorts of things: structure, transport, enzymes, communication. Basically, your proteins are the most important thing in your body. Some examples: sucrase, that's the enzyme that's going to break down sucrose into glucose and fructose. Okay, so an enzyme will help lower the activation energy. When we talk about enzymes a little bit more, we'll get into this. It'll lower the activation energy and allow this reaction to take place. Amylase, okay, that breaks down starch. Hemoglobin is the protein that's in your red blood cells that carries oxygen. Keratin is the protein that's in your skin. So there's a lot of different proteins, and they do a lot of different things. And we're going to spend a lot of time this year talking about different proteins, but this is just an introduction. When I was talking about the different types of amino acids, you can see how they have that central structure. Okay, all of them have this alpha carbon, they have the carboxyl group, the amino group, the hydrogen, and they have a variable R group, and this is what's going to make them different. There are 20 different types of amino acid, and many of these you have to get through your diet. A couple of them you can actually synthesize in your body. And these are going to impact the way a protein structurally functions. And we're going to talk about that. We're going to spend a lot of time talking about that. But I just want you to note that they're a little bit different. Whether they can dissolve in water or not is really dependent on the R group that's attached to them. So some of them have, like you see here, these are just carbons, okay? So carbons and hydrogens, which is very nonpolar. So not going to dissolve in water very well. But some of them have oxygens, okay? So if they have oxygen, they have some electronegative properties there, and they're going to be slightly polar. Okay, here's another group that has some oxygen that's going to be slightly polar and dissolve in water pretty well. Remember, like dissolves like. Elements: carbon, hydrogen, oxygen, and nitrogen. Okay, so that's the third one. And occasionally sulfur. And it's only found in cysteine. If I can, yep, right here. You can see the sulfur right there. Next group, nucleic acids, monomer nucleotides. We're going to spend a ton of time talking about nucleic acid, which are basically DNA and RNA structure. They're genetic material. The linkage between them, so here's the one, a nucleotide with a 5-carbon sugar, a phosphate, and a nitrogenous base. To link two of these little guys together, you're going to use a phosphodiesterase linkage. And then for DNA in particular and RNA during transcription, you're going to note that there are hydrogen bonds. Okay? DNA is double-stranded, okay? And to hold those two double strands together, you're going to use these hydrogen bonds. Okay? And we're going to spend a ton of time talking about that, so just get the basics down today. Elements, okay? There's nitrogen, there's carbon, there's oxygen, there's phosphate, there is hydrogen. The new addition here is the phosphate group. That's not really seen in any of the other groups. This will dissolve in water. Phosphates are very negative, so it's going to have that characteristic of interacting very well with that water. Okay, so let's look at the last group, lipids. Lipids technically do not have monomers because you can't, there aren't repeating subunits, but they do have building blocks. So glycerol and fatty acids, okay, are their building blocks. The purpose, okay, are to store energy in the glyceride form. Okay, this is long-term energy. So if you have any, like fat cells in the body, that's where like the fat is stored for a long period of time before it's needed. Phospholipids are going to be part of membranes. So typically the elements that you'll see are carbon, hydrogen, and oxygen in fatty acids. In phospholipids, though, they will have a phosphate group. Okay, so here's a triglyceride. You might have heard of those. They have 3 fatty acid chains held together with a glycerol molecule. Okay, and then a phospholipid has 2 fatty acid tails and then this phosphate polar head. What's really interesting about these guys is that they're part polar and they're part nonpolar. Okay, so their tail nonpolar, okay, but the head is polar. So this is why they're really great for membranes. And we're going to spend a lot of time talking about membranes. So just note that they're in membranes. Fatty acids, though, okay, or if they're in a glyceride form like this, note that they have a lot of carbon and hydrogen, so they're actually very nonpolar and will not dissolve in water. There's different types. You may have heard of saturated versus unsaturated. Saturated means that there are no double bonds. Okay, they are completely saturated with hydrogen. Unsaturated, on the other hand, have these little kinks, okay, where there's double bonds. And because of those little kinks, they're actually going to be liquid at room temperature, unlike saturated fats, which are usually solid at room temperature. So if you think about something like butter, at room temperature, butter has a lot of saturated fats in it, so it's solid. Something like olive oil is liquid at room temperature. Okay? It has a lot of these kinks in it. This is your introduction to organic molecules. Please bring any questions that you have with you, and we will do a little review and discuss all of the organic molecules in class. Have a nice day, girls.